DNA is made up of molecules called nucleotides. These nucleotides are made up of a phosphate group, deoxyribose, which is a sugar, and a nitrogenous base. There are 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. Adenine is paired with thymine by a double hydrogen bond, and guanine is paired with cytosine by a triple hydrogen bond. These nitrogenous base pairs make up the rungs of the ladder, while the phosphate group and deoxyribose make up the backbone. The nitrogenous bases are held together by a weak hydrogen bond because they constantly need to be pulled apart in order to be copied to make RNA. DNA is shaped in a double helix, which is virtually a twisted ladder. The 2 strands of DNA are antiparallel, which means that they run in opposite directions.
Transcription occurs in the nucleus. There, RNA polymerase, an enzyme, clings to DNA and breaks the hydrogen bonds between the bases to unzip the 2 strands of DNA. Then, mRNA, or messenger RNA, begins to form by complementary bases of one of the strands of DNA contributing to a single other string of nitrogenous bases. These bases create the mRNA strand. The one exception is that instead of thymine being put into the strand as the complement of adenine, uracil is used. RNA polymerase stops making the mRNA when the gene is complete. RNA polymerase knows when the gene is complete because some sets of bases code for a stop signal. No more bases will be added to the mRNA strand, but it is not yet ready to travel out of the nucleus. The mRNA can't go out of the nucleus until it gets edited. The mRNA, carefully copied from the DNA, is coding for a specific gene, but there are extra pieces in the mRNA that are not needed. The mRNA has pieces in it called introns that are not needed for when the mRNA goes out to the ribosome, so these pieces are cut out of the mRNA and left in the nucleus. The mRNA now only has a cap at the front, the exons, or important parts, in the center, and a poly A tail at the end. The mRNA strand is now ready to go out of the nucleus and into the cytoplasm where the ribosome will meet it.
Translation is the process in which mRNA is read and transformed into proteins. Translation occurs in the ribosomes. The mRNA enters into the ribosome, and in the ribosome, the codons, which are sets of 3 nitrogenous bases, are connected with their corresponding anticodon. The anticodon is the opposite of the codon and is part of the tRNA. The tRNA also carries an amino acid. Once in the ribosome, the tRNA is lined up with the codon that is opposite to its anticodon, and as a result, drop their amino acid, which connects itself with the amino acid from the previous tRNA. This amino acid chain that builds up is called a polypeptide chain. When finished, this chain is ended and folds up into a protein. Something important to note is that a given codon will only code for one amino acid. However, there are multiple codons that will result in the same amino acid. There are 4 stages to the structure of proteins. The first degree of protein structure, known as the primary structure, is just the order that the amino acids are arranged into by the mRNA. This order determines the shape of the protein. The second degree, secondary stage, is when the proteins become an alpha helix or a beta-plated sheet because of certain attractions that some amino acids have towards each other. The tertiary stage, and in some cases the final stage, is when the hydrophobic amino acids fold to the interior and the hydrophilic amino acids fold to the exterior. The quaternary stage only occurs with some proteins, but is when multiple polypeptide chains interact to create a protein.
So why are proteins important? Well, we are made out of protein. Proteins are enzymes that help functions go smoothly, as well as transporters, regulators, and defenders. Protein synthesis is important because it is the making of you using your unique, one-of-a-kind DNA code. This process is called the central dogma of biology because biology is the study of life, and proteins are the basis of all living things.